I want to, sometimes I run out of time at the end and I don't get to thank the people and acknowledge the people I want to. So I just want to start out by thanking Tammy and Sarah for putting together today's meeting. The talks this morning were wonderful and it's really great, thank you. As well as NIAAA for supporting this meeting, as well as my research program, NIDA, Rutgers, the Center of Alcohol Studies. The research I'm talking about today is completely team science, and so I wanted to just acknowledge in particular a couple people in the lab.
Is it on?
If I can. No, it was not on. Thank you. Good hint. It's still not on. Evgeny and Branya Vestillo, who are our physiologists we've been lucky to have in the lab for the past 8 years. Jen Buckman, a neuroscientist. Our neuroimaging collaborators, Bharat Biswal from the New Jersey Institute of Technology and Steve Hansen from the Rutgers Brain Imaging Center in Newark. And our computational modeling collaborators from UC Santa Barbara, Igor Mezic and Maria Fonabarova. So the questions that our lab is interested in is how do the brain and the heart work together to determine how we experience emotion, arousal, and stress, and then importantly, what roles might the system play to either enhance or reduce the likelihood of inflexible behavior, especially towards alcohol and other drugs. And so the thing that we've been most interested in recent years is heart rate variability. And variability and complexity are hallmark characteristics of a lot of biological systems, and heart rate variability follows that same principle. So it turns out that the more variability you have in the time intervals between the beats of the heart is actually an incredible indicator of both your physical health and your ability to adapt in everything in the world around you. We started out just adding HRV into a study as a biomarker of physical and mental health, but very quickly became interested in its potential as a physiological process, not just as a static biomarker, but as a process that's dynamic and that unfolds in real time in the same way your brain reacts to things in the world. The neural system that controls heart rate variability is the central autonomic network. And this network operates through the Baroreflex Loop to, at various levels of brain organization, to control the regulation of heart rate and blood pressure. If you've thought about the Baroreflex Loop at all, it probably is just thinking about it in terms of blood pressure control, which it does. It keeps us alive. We don't have strokes, that sort of thing. But it's much more than that, I think you'll see as the talk goes on today. This Baroreflex Loop, as I said, it's a negative closed loop system that connects the brain and the cardiovascular system, and the bodily part of the system is represented by the baroreceptors, which are mechanoreceptors that are in the stretch walls of all the vessels in the body. Some of the most important ones are on the sinoatrial node of the heart and the carotid artery. And through this loop, there is continual fine-grained signaling and feedback that takes place between the body and the neural systems that control these bodily functions. We, over time, have become very interested in the afferent stream of this feedback loop. That is the nature of the feedback that goes from the body to the brain. It's something that hasn't been considered very much, especially with respect to mental disorders and addiction problems, but ends up being a very powerful stream to think about because there are a number of interventions, such as exercise, meditation, heart rate variability, biofeedback, resonance breathing, that all seem to operate through this afferent stream. So manipulating behavior at the periphery can be fed back to control or to modulate activity in the brain at different layers of organization. The sensitivity of this baroreflex loop is increased by all these different interventions through multiple different mechanisms. The particular mechanism we're interested in, or intervention we're interested in, is resonance breathing. That's breathing at the resonance frequency of the heart rate baroreflex loop, which is about 0.1 hertz. And it turns out that this resonance breathing actually is one mechanism. It's the main mechanism that operates in heart rate variability biofeedback, but it's also been found in various forms of meditation and yoga, and some things that I'm going to be talking about a little bit more in a moment. So, over here on the right is the central autonomic network, and I'm sorry, this is kind of a fuzzy slide, but I just wanted to point out a couple things involved in the central autonomic network that regulates heart rate and blood pressures: medial prefrontal cortex, anterior cingulate, insular cortex, extended amygdala, hippocampus, down through areas in the midbrain, the pons, and then important centers in the brainstem, especially the nucleus tractus solitarius and the nucleus ambiguus. And as you can see in this diagram, the layers of feedback between all these levels of organization in the brain are bidirectional. A curious thing has kind of happened over the years, when oftentimes you see a figure similar to this, is that the bidirectional feedback from the cardiovascular system to the brain has often disappeared on the pictures, kind of flying in the face of about 150 years of physiological research in animals and humans. And so the feedback is really bidirectional at all levels, and as I said, this afferent stream is particularly interesting in terms of thinking about ways that we can behaviorally modify systems in the brain. So what we're interested in doing is starting to elaborate some of the predominant addiction models that exist today, in terms of taking it from the brainstem to the body and recognizing that there are these bidirectional paths of influence, and that if we can influence those paths in the right way, we may be able to have an effect on reward circuits in the brain, potentially interrupting overlearned conditioned responses that contribute to stress reactivity, negative affect, and craving, and potentially also strengthen regions of the brain involved in executive functioning. I think an important point is that the central autonomic network that controls heart rate and blood pressure is not a parallel or independent circuit from circuits that control affect, emotional response, craving, addiction, and stress. And so these are major integration relay parts of the brain where information about physiological processing is integrated with cognitive emotional information as well. So as I said, heart rate variability is simply a variance in the time intervals between heartbeats. And when I first heard that, I was surprised. I had thought if you had a perfectly regular heartbeat, that would have probably been the healthiest way to be, but you would actually be dead if you had no variability between the intervals of your heart rate, so heartbeat. So I was incorrect on that one. But there is substantial bodies of literature, as I said, considering heart rate variability in the resting state as a biomarker. Lower levels of heart rate variability are associated with all-cause mortality, poor physical health, poor mental health, poor immune response, depression, panic. The list goes on and on, hundreds and hundreds of articles. And the alcohol area has actually in recent years gotten somewhat interested into this, construct of heart rate variability. These are two recent review articles that were published in ACER talking about the effects of chronic heavy alcohol use in terms of lowering heart rate variability and as well acute effects decrease heart rate variability. So what we're really talking about is alcohol interrupting these really fine-grained adjustments that take place between heart rate and blood pressure. The decrease in heart rate variability is caused probably in large part by negative effects on the baroreflex loop that are both centrally and peripherally mediated. So alcohol increasing sympathetic arousal, decreasing vagus nerve activity, negatively affecting blood vessels, and overall dampening the sensitivity of the system to react to change. So our initial studies of heart rate variability as a process was really looking at differences between individuals in terms of their reactivity both to alcohol and to various kinds of challenges, for example, emotional challenges, understanding how heart rate variability changed in different people in response to positive and negative cues. Also looking at gender differences in the acute effects of alcohol in a couple of studies. And I thought one thing that was interesting in a slide that we saw in the opening session was, I think it was in developing countries, there was a gender difference, I believe, in negative physical health problems in developed countries, with women showing substantially less than, girls showing substantially less than men. And I would hypothesize that one aspect of that is the cardiovascular excellence of women, if you will, at all ages. And as well, in respect to alcohol, acute alcohol, it disrupts heart rate variability in women as well as men, but to a much less extent. And we also showed that during acute intoxication, that the adaptive redistribution of heart rate variability in the spectral density function towards low frequency is much greater in women than men. And so there are other aspects of alcohol that seem to be more deleterious to women than men, but it seems like the cardiovascular system may be a particularly resilient system in women that might be useful, as I talk later about intervention effects that may be particularly good in women. We've shown that heavy binge drinkers in college show much more heart rate reactivity to both alcohol and drug cues than lighter drinkers. Different associations of reactivity with cognitive expectancies about alcohol use, and also over time in heavy drinkers, a growing disconnect between physiological arousal responses to emotional and alcohol-related stimuli compared to individuals' subjective reports of what their level of arousal is. So it's showing less integration with perceived levels and physiological levels. But probably the most important thing that I want to talk about is more about the power of heart rate variability and the baroreflex loop as a modifiable process. And so our more recent work has really started to look at other literatures and develop models of how behavioral interventions to improve sensitivity in the baroreflex system might actually be used to increase individuals' behavioral flexibility towards alcohol and drugs. We primarily are looking at resonance breathing, which I'm going to talk about in a lot more detail, but you can also stimulate or generate resonance in the system by using rhythmical muscle tension at 0.1 hertz as well. It's not as strong an effect, but it's there. You can influence the very low frequency part of the spectrum, which has to do with vascular aspects of the cardiovascular system with rhythmical sighing. And then in our initial study of a brief heart rate variability biofeedback, we found a medium effect size greater decrease in craving in young adult men who were in 30-day inpatient treatment for alcohol and drug use disorders. Didn't quite reach statistical significance. They were in an intensive program everybody's craving reduced, but we did, in this relatively small sample, have a medium effect size greater reduction. The standard course of heart rate variability biofeedback involves about 10 one-hour sessions of training and practice with a therapist, and then practicing 20 minutes twice a day on your own. Basically nothing anybody would ever do in an addiction program. What we tried here was just giving 3 sessions, asking people to practice. Many of them didn't, which probably also decreased the effect. If you look at the literature, it's interesting that not only in heart rate variability biofeedback, but 0.1 Hertz resonance breathing appears to be a common mechanism across several different forms of meditation as well as breathing yogas, where studies that have been done have found that individuals are spontaneously starting to breathe at 0.1 Hertz during the practice. It's also been found for certain chanting and saying the rosary, although according to the British Journal of Medicine, the Hail Marys have to be said in Latin. You have to do the Ave Marias, where you don't get the resonance effect. So what's happening in the heart rate time series when you do a spectral analysis during resonance breathing? And in general, during resonance breathing, you have a massive increase in heart rate variability, which is very healthy. But you see by this amplitude at 0.1 Hertz that almost all the increase in variability is a buildup in the system in the very low frequency, or in the low frequency range, exactly at 0.1 Hertz, where the high frequency, which is a purely parasympathetically mediated portion of the spectrum, you don't see really much activity. You see a little bit here. It's a combination of vascular response and noise. For those of you who might have an easier time thinking about breathing in terms of the time domain instead of the spectral domain, resonance breathing is doing approximately 6 breaths per minute, so taking about 10 seconds for one complete inhalation, exhalation. Usually people are advised to inhale for about 4 seconds and exhale for 6 seconds. It's substantially slower than the normal breathing rate, which is around 15 to 17 breaths per minute. You have to just be careful not to breathe too deeply or you'll hyperventilate. But if you avoid hyperventilating by breathing into your stomach and breathing slowly but not too deeply, basically what you start to feel almost immediately is just incredibly relaxed and nice, especially if you have a lot of sympathetic activation going on, and just dampen it right down. In general, though, it's not just a way to dampen sympathetic activation, but more to create balance between the sympathetic and parasympathetic branches. So in normal populations, after a course of heart rate variability, biofeedback, or resonance breathing, there are actually chronic gains chronic increases in baroreflex gain. In clinical populations, you get substantial clinical benefits, that is, decreased symptoms of depression and stress and anxiety. All the clinical indicators are good, but you don't see or the studies thus far haven't seen these long-term chronic physiological effects, which suggests to us that it may be that short epochs of resonance breathing may work clinically by inducing a positive change very close in time to when you're breathing, and that the more that you do that, the more likely you are to have the sustained clinical benefits. And if you do it often enough, it anecdotally, in many of our participants, almost becomes an automatic response when you're in a stressful or anxiety-producing situation, that you just slow your breath rate. It used to require a long time in the lab and an EKG and a lot of sensors and doing a lot of things to assess heart rate variability and to provide biofeedback. But we're just starting a new randomized clinical trial where we have an iPhone app that we downloaded. We kept switching which one we're using. I think this might be training for HRV. But in any case, you put your finger over the camera, which acts as a plethysmograph. It shows you your heart rate trace, and it provides a small pacer, which is right there. And essentially, you're just told to inhale as the pacer goes up, and exhale as the pacer goes down. The idea in our study is that we're giving it to women, women who are parenting young children who happen to be in alcohol and drug use intensive outpatient treatment, and we're training them how to do the breathing very briefly, and asking them to use it just as another tool that they have in the moment whenever they anticipate or experience a trigger in their day-to-day life. So it's something that you can take out of the lab or the treatment context and use during your day-to-day experience, which is when relapse and those sorts of things happen.
Can I just ask, how's it estimating the respiration?
It doesn't estimate respiration. There's a pacer that you set the timing of the pacer.
But there's a respiration trace on there.
Oh, that's a heart rate trace that's getting from pulse in your finger by putting your finger over the camera.
Red, but the blue line.
Oh, I'm sorry.
Blue line is the respiration.
Yes, so these lines don't actually correspond to the data from that app. You can get the heart rate data from the app. We're downloading it once a week. We're in the process of studying, I think it's reasonably good how close, because they're walking around and moving and talking and doing all these kind of things. So we have to look at how closely it really approximates in laboratory types of assessments.
And you're coming up with the HRV, feeding that into a program somehow, or does it give you HRV measures?
It gives you, when you dump the data, it'll compute a few HRV measures for you, but we're taking the time series to look at, yeah. And again, so the left side of the figure is just breathing at a normal frequency, and the right side of the figure is breathing at resonance. And you can see that in addition, the middle rectangle is the RR interval from ECG, but you can see that you're also inducing high amplitude oscillations in arterial pressure, and you're also tremendously increasing baroreflex sensitivity as measured through baroreflex gain. So one direction we've decided to go with this is resonance breathing, just like any other intervention, is not going to work for everybody. Some people aren't going to have difficulties modulating arousal to begin with. Other people will, but the intervention won't be effective. And so we, with our computational modeling colleagues, developed a computational model of the baroreflex with respiration as the only input, and was actually based on substantial work that was done in dogs, where they do most of this research for some reason. And we were able to very closely reproduce the heart rate spectrum, both at baseline, where you can see the mass of power in the high frequency domain, which is parasympathetic, and then during resonance breathing, where the power is at 0.1 hertz. The idea with the computational model is we're enabled to include about 90 underlying physiological parameters. We were able to identify, I think, about 7 parameters that really change substantially between resonance breathing and regular breathing. We can plot those out at the individual level, and I'll show you just an example here. This CSP is a measure of increased blood flow to the internal organs. So we can look at, through computationally, a lot of things that you couldn't look at. You would have to be invasive to look at, or wouldn't be able to look at all. So these are 2 baseline conditions. You can see that during resonance breathing, that there's a great increase in blood flow. You can also see from these error bars that there are a lot of individual differences in the extent to which people experience that positive effect. And below, this is the modal response. So if you just look at the mean, individuals this is the first baseline, and they become a little bit more relaxed here, and then when they start breathing at resonance, there's this huge increase in blood flow. But you can see that about a quarter of the participants don't experience that positive effect. They experience atypical things or actually a negative effect from it. So right now what we're doing is working on a computationally efficient form of this model so that you would be able to take a 5-minute ECG trace and respiration and run it through the model and actually have a prognostic indicator of which individuals you would anticipate would positively benefit from the resonant breathing intervention, not just on the basis of this parameter, but on the basis of the 7 parameters that we identified. So then life was good until we started to take our paradigm into the MRI environment to see what was happening in the brain at the same time. And I just put this slide up for a moment. Despite what BioPack and everybody else tells you, it is not easy to get heart rate data in the MRI once you turn the magnet on. It's just, so you can do all their frequency band, steps to remove noise. But if you can just, let's see, this is the functional scan on, this is the scan off. This is blood pressure, and the blood pressure signal doesn't get degraded at all. It's quite wonderful, beat-to-beat blood pressure. But you can see, these are pretty clear R spikes, but then you have this area of the tracing where it's not clear what's going on. And after the corrections, it's still not clear what's going on. But if you assess beat-to-beat blood pressure at the same time, you can use the amplitude and the relationship between blood pressure and heart rate to actually be able to pull out where the R spike is. Without doing that, we would lose data on about at least half of the subjects. There are huge individual differences also. Some people in the magnet have lovely tracings, and some people it's impossible to get a signal from. And the ones you can't get signals from are usually women, for whatever reason. Okay, so in terms of an initial study that we did for, demonstration study, really, it wasn't quite clear whether people would be able to effectively do resonance breathing in the magnet because of, what a horrible environment it is. But in fact, they were able to, and you can see this huge build up in oscillations of low frequency heart rate variability during resonance breathing. No real change in high frequency heart rate variability, which is the same thing you observe in the lab. In terms of the brain, we found significant activation of the brainstem and the cerebellum during slow-paced breathing. It's a very small sample. And then we also found significant deactivation of the medial prefrontal cortex also during breathing. So the brainstem activation, was expected. It's the central integration point where the body is sending signals to the brain. And, it's from that point that it gets relayed to the midbrain and higher order structures. The cerebellum isn't part of the CAN. However, the effortful motor control of breathing activates the cerebellum, so that's expected also. Medial prefrontal cortex was also affected by the changes, although it was in the direction of deactivation. And, in thinking about that change and recognizing that medial prefrontal cortex is part of the reward network, it may be the case that activation of medial prefrontal cortex, when you don't have co-occurring limbic activation, may be what's in part related to, did I say activation? I meant deactivation of medial prefrontal, may underlie subjective reports of relaxation and stress reduction, and we currently are doing larger studies so that we can look at some of the behavior-brain relationships. And then the last data I really want to show you, and I thought this study would be completed at this point in time. However, we were recruiting groups of young, non-treatment seeking alcohol dependent subjects and low-risk drinkers. And it turns out in New Jersey that non-treatment seeking alcohol dependent men and women between the age of 18 and 24 all at least smoke marijuana on a regular basis. So we started the study looking for the squeaky clean group, and we will finish it, but we've screened about 450 individuals to get 12 in the alcohol group. So anyway, the study's not done, but it will be done eventually. As it's clear, the samples are racially diverse and well matched, with the exception of the drinking. These are standard drinks in the past year and day, and varies dramatically between the groups, as well as alcohol problems and craving. Basically, we bring them into the imaging center. We train them either in resonance breathing or a task called the vanilla task, which is just a very low demand cognitive task. You see rectangles of different colors float across the screen, and at the end we tell you to silently count the number of blue ones. So it's keeps people looking at the screen, but it doesn't really require much effort. For structural scans, we have 5 tasks, each 5 minutes each, 30-second interval between the tasks where we ask about craving. We have neutral cues, and I should say our 5-minute tasks are rather long for fMRI tasks, and what we decided, or what we've been doing in this initial study, is trying to optimize the cardiovascular assessment portion, because we are bringing the imaging into that program. So there's some things about our design that I'll comment again on later, but there's always difficulty in integrating paradigms from 2 different levels of study. During the nature cues, we just ask them to press a button anytime they see a flower to maintain their attention on the task. The alcoholic cues, we ask them to press a button when they see their preferred drink. And our alcohol cue pictures were actually modified based on the individual's preferred drink, so that we loaded the set with what they like to drink the best. And it's just an example of the neutral and alcohol cues, which were matched in terms of having people in the pictures and complexity and that sort of thing. We presented the cues also at 0.1 hertz, so the picture was on for 6 seconds and off for 4 seconds. And basically, this is just a preliminary analysis looking at the contrast of activation to the alcohol cues presented the second time greater than the first, alcohol cues presented the first time greater than the second, with the resonant breathing condition greater than the plain vanilla condition. So resonance breathing and vanilla task were 2 different groups of subjects who were randomized, and the alcohol cues were within. Basically, the second time that the alcohol cues were shown, we observed more activation in the medial prefrontal cortex, the cingulate, the precuneus regions, the brainstem, and we observed more so in the resonance breathing group than in the vanilla group or the control group. And we observed reduced activation the second time they saw the cues in the visual regions of the brain. Again, more so in the resonance breathing group than in the control group. So just, some, again, very preliminary hypotheses based on these results is that the prefrontal deactivation that we observed during resonance breathing seemed to be potentially followed by a rebound in medial prefrontal cortex activity in response to the alcohol cues, and that this was accompanied by reduced visual processing of the cues. So in terms of the dual process model, medial prefrontal cortex activation is usually looked at as signaling reward value, but potentially again in the absence of limbic activation and with reduced visual processing of the cues may in fact, it may be important to interpret the context in which the medial prefrontal activation is occurring to understand its meaning. Some alternative hypotheses, it's not likely that the reduced visual processing was identified due to boredom because it varied across the two experimental groups, which we assume we bored equally. We also considered whether the changes in BOLD could be affected by changes in pH that occur during resonance breathing. The changes in pH are observed throughout the brain, and we only see changes in certain areas, so it's not consistent with that idea. I wasn't able to, in these analyses, separate the alcohol and the alcohol dependent and the control group when we looked at changes due to breathing, and that's obviously something that we need to do when we have more subjects. And other analyses that need to be done, of course, are including covariates in the model to understand behavioral associations, as well as looking at connectivity analyses between the visual processing areas and the reward and salience regions. And then finally, to elaborate our computational model to begin to include the neural component of the system. So just ending up with some kind of big, picture considerations. In the fMRI area in general, it's continually talked about the need to differentiate the neuron-specific effects from basic physiological functions that are happening in the brain, things related to vascular changes, blood flow that are not related to neural activity. But at the same time, and that's important, there's a tension between, quote, controlling for respiration, just considering it an artifact that you want to get rid of, versus trying to understand the aspects of respiration that are related to differences in neural activation versus those that are more mechanical and not. And it's a very different way of looking at it, which I think will lead, we need better conceptual models to separate those two things. And also more work needs to be done in terms of optimizing designs that would allow for simultaneous physiological heart rate and blood pressure assessment at the same time that we're collecting BOLD signals. Because using these cues in the past, we have seen limbic activation and insular activation that we did not see in this study. So could just be a different sample, could be related to the time parameters we chose in the imaging portion. Thank you.